Hi everyone, we will be discussing research related to the question, what is the mechanism by which the tumor suppressor protein BRCA1 acts as a proapoptotic protein? BRCA1 is a large protein often recognized for its role in maintaining genome integrity by repairing double-stranded DNA breaks via homologous recombination. BRCA1 is also a nuclear cytoplasmic shuttling protein. As a tumor suppressor, systolic BRCA1 was hypothesized to interact with IP3R to facilitate apoptosis. IP3 receptors are expressed on the endoplasmic reticulum and mediate calcium transport, which plays a vital role in cancer. As such, numerous proto-oncogenes and tumor suppressors regulate calcium transport by interacting with IP3R. Excessive calcium flux from the ER to mitochondria results in loss of membrane integrity and influences proapoptotic pathways.
The following paper identifies BRCA1's role in promoting apoptosis through increased calcium release via its physical and functional interactions with IP3 receptors. In this paper, the authors show that there is a direct protein-protein interaction between BRCA1 and IP3R that occurs between the ring domain of BRCA1 and the tail domain of IP3R using a pull-down experiment shown in panel A. Interactions between full-length BRCA1 and IP3R in cells were also confirmed by successful co-immunoprecipitation of endogenous IP3R1 with endogenous BRCA1 in HeLa cells, evidenced in panel B. The authors also investigated subcellular localization of BRCA1 using differential centrifugation. As illustrated in panel C, the authors found that non-nuclear BRCA1 is localized to the same subcellular compartments as IP3R, which together with BRCA1's interactions with IP3R suggests its potential role in modulating IP3R activity. The authors then demonstrated that BRCA1 does indeed regulate IP3R function and calcium release. Using Cura2, the authors measured cytosolic calcium levels in response to different levels of histamine in cells with or without siRNA-mediated BRCA1 knockdown. In response to 100 nanomolar histamine, cells with BRCA1 knockdown had lower peak release and a fewer percentage of responding cells compared to cells expressing the control siRNA. And in response to 1 in 10 micromolar histamine, cells with BRCA1 knockdown had decreased oscillation frequency and a lower peak release compared to the control, indicating that BRCA1 significantly affects IP3R channels in cells' calcium release profiles. The authors further demonstrated that endogenous BRCA1 increases calcium release by directly modulating IP3R channel gating and specifically increasing the open probability of the IP3R channel, as seen in panel A and quantified in panels B and C.
This figure accompanies the hypothesis that BRCA1 binding to IP3R increases during cell death and that this is an essential component of its tumor suppressor activity. FRET was used to pair CFP IP3R and YFP BRCA1 to measure the changes in their interaction during cell apoptosis. Comparing figures 5B and 5C, there is a significant increase in FRET within the cells expressing the pair compared to expressing only one of them. In figures 5D through F, a mutant BRCA cell line, which silences BRCA1 protein expression, was compared to a wild type to view its effects on cytosolic calcium levels. The high correlation between the wild type and the significantly elevated calcium levels indicates that BRCA1 protein production is required for apoptotic calcium release through the IP3R, which is also consistent with the cell death results shown in figure 5F.
Using adaptive blasts, the authors identified the lipid binding region within the BRCT domain shown in the bottom panel. The region has a strong potential to lipid binding due to the homology of fatty acid binding proteins. Highlighted in purple are solvent-accessible residues in the basic region of BRCT1 domain, potentially indicating lipid binding pocket activity. Finally, the authors purified this recombinant BRTC domain to perform lipid stray overlay experiment. It is clear that BRCT domain could bind with several membrane lipids evidenced by dark spots, whereas the negative control, the ring domain, did not. This proves that BRCA1 has a lipid binding domain associated with the ER, which may allow rapid recruitment to IP3R during apoptosis.
This paper highlights the significance of the systolic proapoptotic function of BRCA1 in relation to both hereditary and non-hereditary cancers. Downregulation of BRCA1 may be present when cancers without BRCA1 mutations. Correlation of systolic BRCA1 with a better disease prognosis then suggests that the proapoptotic function of BRCA1 has significant implications in mediating cellular responses. Research is also needed to understand the role of calcium homeostasis in tumor progression and how regulators of IP3R like BRCA1 may be useful in developing cancer therapies.